Hello, today we are going to be talking about the structure of DNA as well as the replication process that goes along with it. So why DNA is so important and why we talk about it is that it is absolutely essential to life. It stores information about the way we are, it's passed down from our parents, it controls the production of protein, which is another process that we'll talk about, which helps tell cells what type to become. So that's that cell specialization we've talked about. And it is absolutely unique and individual. So it helps to identify people. So I want you to pause the video and write down at least one circumstance that you could see having unique DNA being helpful in. Some different circumstances could be the identification of a perpetrator, maybe. Others could be identification for documents, and I'm interested to see what else you guys come up with. So the history of the discovery of the DNA double helix, which is what you guys watched a video on this weekend. The first person to be involved is someone called Frederick Griffith, and he discovered that there's a factor in disease bacteria that can transform harmless bacteria into deadly bacteria. So something was being passed along, and that was in 1928. In 1952, Rosalind Franklin, who you now know had her work stolen basically from her, took an X-ray photo of DNA that was really, really good and led Watson and Crick to be able to describe the DNA molecule in 1953. So in summary, for this part, genes are units of genetic material that code for a specific trait, also called nucleic acids. And then we have these things called nucleotides that are repeating molecules that give the sequence of DNA. Here you see one nucleotide, so I believe I gave you an area that you could draw. Don't worry about these letters hanging out here. I would draw a circle. This is a phosphate group. This house-looking thing is a sugar. It's called deoxyribose, which is right here. And then you have something called a nitrogenous base. And I'll show you in a second. These make up the backbone of DNA, and these make up kind of the middle portion of DNA. There can be four different nitrogenous bases, A, G, C, or T, and we'll get more into those in a hot second. But I would go ahead and draw and label all three of these components. This whole thing is considered a nucleotide. So what do all these words mean? Phosphate group is the backbone supporting structure of the entire molecule. Deoxyribose is the sugar. That's the five-sided structure. It's attached to the bases. It's also a portion of the backbone. And then you have the nitrogenous bases, which are the rungs of the ladder, made up of potentially A, T, C, and G. So Watson and Crick proposed that there were specific pairings between the nitrogen bases. A, or adenine, always pairs with T, or thymine. C, cytosine, always pairs with G, or guanine. Highlight, underline, circle, everything that. DNA was made of two long strands, that's super important, arranged in a way called the complementary rule. So if you have an A on this side, you would have a T on this side of DNA. So here's where the double helix happens. Double meaning two, helix meaning spiral. The legs of the ladder, so this would be before it's twisted, is the phosphate sugar backbone. And then the rungs of the ladder would be the bases, A, T, C, or G. In real life, it looks like this. And I would go ahead and draw both of these in your notes, and I would label them how they're labeled here. So this is an actual representation of DNA. These are your sugars, phosphate, another sugar, and you have a base attached to it. Phosphate, sugar, base attached to it, and you see you have a cytosine or a C here, and G is attached on the other side. Here you have an adenine, and you have a thymine attached here. So these bases can be put into categories. The first one is something called a purine, and that is either A or G, and these are two-part. So you have this part and this part, whereas this one down here is only one part. So if you look here, a purine, adenine, pairs with a pyrimidine, which is T. Guanine with cytosine. So you have a two-parter attaching to a one-parter. Chargaff's rule. So Chargaff actually put into play that A must pair with T, G must pair with C. So their amounts in a given DNA molecule will be about the same. So if you have 36 A's, you're also going to have 36 T's. If you have 42 G's, you're also going to have 42 C's. In the middle here, there is something called an H bond. This is the bond that holds the two bases together, and that is a hydrogen or an H bond. It's a very, very weak bond that holds them together, but can also be broken when we go through the replication process. So what I'd like you to do is take a hot second, and I want you to create the complement to the other part of these DNA strands. So here you have an A, underneath you would write a T. Here's a T, underneath you would write an A, G. So go ahead and complete those, and I will go over those once everyone is done with the lecture for today. It's super important to have these different arrangements of nucleotides because it provides diversity among living organisms. The specific order that the bases occur in is the code of life. And then to start talking about replication, replication means to copy, make an identical part to fit into our cells. DNA is wrapped tightly around something called histones, and that squishes them together so that they are able to coil tightly and form chromosomes. And here's the first thing that happens. This is your strand of DNA, and what we've done is we've gone bloop, and we have broken the hydrogen bonds. So the bases break apart, and it is something called DNA helicase that breaks those bonds apart. Step 2, you have the strands move apart farther. Step 3, we now base pair again on both sides. So here you paired this in, this one in. Okay, and on this side you did just the opposite. You paired this dark green with the light green, peach with the brown, light with the dark. So each of the unzipped strands forms a template for a new strand. And then step 4, we now glue the strands back together, and they're glued back together with something called polymerase. So we go bloop, and we glue it back together. Now you have two identical strands of DNA, each one built on an old strand here and an old strand here, and then you have the new strands in the middle. Okay? And that is called the semi-conservative model, and I would go ahead and write that down. And this is just some quick facts. DNA from a single human cell can extend for almost 2 meters long, and it has the information equal to 600,000 printed pages of 500 words each. That's a library of about 1,000 books. So I would like you to hop back on Google Classroom, watch this DNA replication video, and then get out your take-home or your crossword to work on before we cover this information in class together as a group.